and she will talk about the genome-wide association studies of MRI hyperintensities in a small artery disease of the brain.
Thank you very much, Professor Ménard, for the kind introduction. And I would like to thank very much Professor Marie-Germaine Bousser and the Fondation Singer-Polignac for the very kind invitation in this prestigious environment. So I will be talking about genome-wide association studies of small artery disease of the brain, and we'll be focusing primarily on MRI-defined white matter hyperintensities. So I will start with a short introduction to the phenotype I'm looking at and to the method of GWAS. So, as you know, small artery disease of the brain can manifest as acute stroke. It can be acute ischemic stroke, small subcortical ischemic stroke, or it can be intracerebral hemorrhage. However, the burden of small artery disease expands far beyond that of acute neurological events. And in fact, MRI features of small artery disease are highly prevalent in the general population, especially in older individuals, when MRI scans are performed systematically. And so these features, we've been discussing them quite a lot in the previous talks, but include white matter hyperintensity, small subcortical brain infarcts, cerebral microbleeds, and there are other features such as dilated perivascular spaces that I have not shown here. So I prefer to call them covert than silent because they are not completely silent. In fact, it was shown that they're associated with a number of subtle cognitive changes, motor impairment, and they predict a significantly increased risk of stroke, dementia, and death. So among all these, I will focus more on white matter hyperintensities for several reasons. First of all, because the heritability of white matter hyperintensities was shown to be quite high. It was estimated between 55% and 80% in family studies. And besides, in contrast with the other MRI features that I have shown, white matter hyperintensities can be studied quantitatively, which increases statistical power to detect associations. And why study these MRI endophenotypes rather than stroke, for instance? Well, one argument is that there's the assumption that endophenotypes or intermediate phenotypes, such as white matter hyperintensities, could reflect a less complex genetic architecture than more downstream clinical events, such as stroke or dementia. So white matter hyperintensities, as has been also discussed, are indicators of small artery disease, and they can be associated with various features of small artery disease, and actually I should say diseases. So lipohyalinosis, arteriolosclerosis, atherosclerosis at the origin of perforating arteries, and also cerebral amyloid angiopathy, which actually is highly prevalent in older people. And so there are a number of pathological correlates of white matter hyperintensities that I'm not an expert of, so I will not dwell on these too long. But the pathophysiology of white matter hyperintensities is in fact imperfectly understood. They are often considered as areas of incomplete infarction, and as has been shown on very elegant pictures, one of the issues is that there are no anastomoses in the vascular architecture of the deep brain regions, and so this increases the vulnerability of these regions to chronic hypoperfusion. Other mechanisms have been suggested to explain white matter hyperintensities, including dysfunction of cerebral arterioles, for instance, or blood-brain barrier disruption, and I will refer you to the excellent, highly cited review of Dr. Wardlow in Lancet Neurology that has very detailed descriptions of these hypotheses. So a few risk factors are actually known for white matter hyperintensities or have been robustly shown. And to date, age and hypertension are really deemed the most important predictors of white matter hyperintensity burden. And we've talked about, Dr. Norving has talked about hypertension, and midlife blood pressure measurements may be better predictors. But overall, not all individuals with white matter hyperintensities are hypertensive, and other risk factors, and probably some of these genetic, may be involved. So what tools do we have available to try to identify these genetic risk factors? So one thing I should have said right away is that I will be talking only about complex or common forms of small artery disease of the brain and not at all about the monogenic forms. So in the setting of complex forms of disease, the most appropriate tool is to perform genetic association studies rather than linkage studies. And genetic association studies simply consist of comparing the frequency of genetic variance between patients and controls, or across the spectrum of a continuous trait, such as white matter hyperintensity volume. So the first generation of genetic association studies were called candidate gene studies because they were based on a priori hypotheses on the underlying pathophysiology, and the association of genetic variants in so-called candidate genes were tested with many diseases, and in fact several thousands of candidate gene association studies have been published, and they have been quite disappointing because most of these findings have never been consistently replicated. However, this was the early generation of genetic studies, and I think people were in the learning curve, I would say, of genetics of complex diseases, so there were a number of methodological flaws of these studies, which had small sample sizes, they didn't pre-plan replication, they did very limited screening of genetic variation, and of course, if they picked the wrong candidate, then the likelihood of finding something was quite low. So a number of candidate gene studies have been published for white matter hyperintensities, and this list is only genes for which at least one study has published a positive, significant association. And as you can see, in most cases, only one study was published on a given gene, and with just one, maybe two variants. And often these studies did not include replication internally. So in the beginning in 2005 to 2006 approximately, a new generation of genetic association studies became available thanks to the advent of high-throughput genotyping or chip technology, and also to the International HapMap project that provided to the community a detailed catalog of genetic variation across different ethnic groups. So genome-wide association studies do not assume any a priori hypothesis. They use an agnostic approach, and thanks to this high-throughput genotyping, they consist of genotyping very large numbers of genetic variants, single nucleotide polymorphisms, which are basically the most common form of genetic variation that is studied in complex diseases. And so they genotype genetic variants that are distributed all across the chromosomes. And so microarrays comprise between 500,000 up to now about 5 million genetic variants. And in addition to these typed variants, there are now statistical techniques that enable to infer additional variants using these reference panels that I referred to, now the 1,000 genomes reference panels, so that in total about 38 million variants can be studied across the genome in association with any disease. So GWAS have identified thousands of novel loci associated with various traits. So each dot corresponds to, each color is one disease, and these are the chromosomes. And I will discuss this a bit more extensively tomorrow. But I would just like to point out one key point is that genome-wide association studies imply a lot of testing. Multiple tests are performed, so you have to correct for the number of tests you performed, and so this means that to have sufficient power to demonstrate any association, you need to have very large sample sizes. Most of the time, to reach such large sample sizes, you need to work in collaboration with other groups across the globe. So much of this work is being done as part of large consortia. And this was the case, actually, of the first GWAS of white matter hyperintensity volume, of which I will summarize the findings. And this was published, actually, already in 2011 by the CHARGE group, and was led by Miriam Fornage from Texas. And so the CHARGE consortium is an investigator-driven collaboration of large population-based studies aiming at facilitating the meta-analysis of large genome-wide association studies. This GWAS comprised in total a bit more than 9,000 individuals of European descent from 7 cohorts. With a mean age of about 70 years, there was a predominance of women, and more than half of the sample had hypertension. So participants with stroke or TIA were excluded at the time of MRI, but this is a population-based sample, so this doesn't mean many exclusions. And The quantification of white matter hyperintensity volume was done mostly using automated or semi-automated segmentation algorithms, which leads to a volume really in milliliters, an actual continuous volume. But two studies used visual scoring, did not have automated measurements available, and used a visual scoring on a 10-point scale. However, the correlation between this visual scoring and the automated segmentation volumes was actually very good. So I won't go into the details of this slide. I was asked to be a bit faster. So just to say that in total, 2.5 million SNPs were analyzed across the cohorts, thanks to the method of imputation that I referred to before, that enables statistical inference of all the variants present on a given reference panel. And a meta-analysis of the findings identified in each cohort was performed using a special technique that took into account the fact that the phenotype was not measured on the same scale in all studies. And this is a graphical representation of the results. So you've probably seen such plots before. So they're called Manhattan plots because they look like skyscrapers. But so each chromosome, so each color is a different chromosome, and each dot corresponds to one SNP. So the x-axis is the position on the SNP, on the chromosome, sorry, and the y-axis corresponds to the minus log of the p-value. So if you have 6 on the y-axis, it means you have a p-value of 10 to the minus 6. And given the multiple corrections, the corrections for multiple testing, which are done for about 1 million independent SNPs across the genome, to reach significance, you have to reach a p-value of less than 5 times 10 to the minus 8. So one locus reached this p-value threshold on chromosome 17 with a p-value of 4 times 10 to the minus 9. 17q25 is the locus, precisely. And this is a zoom, a very sharp zoom on that region. And as you can see, there are actually several variants that reach genome-wide significance in this region, and they actually, in linkage disequilibrium, they are quite, they're correlated with each other. And we selected for replication the most significant SNP and among all the significant SNPs, the one that was the least correlated with the top SNP, with the underlying idea that maybe these could be tagging to independent loci. So the findings were replicated in the initial publication in 2 independent European population-based samples, the AGES and 3C Dijon study, quite convincingly. As you can see, the p-values were quite significant. So for replication, since you have only 2 SNPs, you don't need the threshold of 5 times 10 to the minus 8. You can be less restrictive. So this is highly significant replication. It was not replicated in a small sample of African American individuals, but first of all, I think we may have lacked power here. And second, it is well known that the allele frequencies and the genetic architecture in general differs between ethnic groups, so it is not completely surprising. Importantly, after this initial publications, there have been 3 completely independent papers that have been published and that have all confirmed this association. So the first one in the generation 2 and 3 of the Rotterdam study, so population-based setting. The second one in the Metastroke study that was referred to before, which is a meta-analysis of GWAS of ischemic stroke, but a subgroup of patients also had undergone brain MRI. And so white matter hyperintensity volume was measured in stroke patients. So it means that this association is also found in stroke patients with white matter hyperintensity volume measured. And finally, it was also confirmed in a Japanese study of healthy adults, suggesting that this association is not restricted to European individuals. But what do these findings mean, after all? So, So the top SNP is located in an intron of the TRIM65 gene and is associated with expression levels of the TRIM47 gene, the nearby gene. And the second SNP we had selected for replication is located in an intron of TRIM47. So TRIM stands for tripartite motif containing. So these are ring finger B box coiled proteins, name. And they're supposedly involved in a number of important processes such as apoptosis, neuroprotection, innate immunity. And interestingly, both of these genes encode E3 ubiquitin ligases of the ring finger type. So the ubiquitin proteasome system was discovered actually relatively recently and targets numerous cellular proteins for degradation. So it's a very important process, very complex process that involves many enzymes, of which the family of E3 ligases. And it was shown to regulate a very large number of basic cellular processes. So I don't know how relevant this is to our findings, but I was quite intrigued by two recent publications that have investigated ischemia reperfusion in animal models, rat or mouse models, and have found that ubiquitination and deubiquitination may play an important role in response to ischemia and reperfusion, potentially modulating neuroprotection and inflammatory response to this phenomenon of ischemia reperfusion. So, I mean, this is just food for thought, and I don't want to, carry the speculation any further. But it's interesting, I think. But I have to emphasize that genetic associations from genetic association studies do not necessarily imply causality. And in fact, most of the time, the associations that are observed just correspond to associations with a marker. The typed variant is often just a marker of the true causal variant, which is not typed, which is probably somewhere in the same region and is in linkage disequilibrium with the marker that has been typed, but is not seen. And besides, so we have seen that the variants we have identified here are non-coding variants, and non-coding variants may be involved in the regulation of transcription of genes, but not necessarily the closest gene. Sometimes it can be more distant genes as well. So in other words, you cannot conclude anything about causal variants or causal genes from results of a genetic association study, and this requires additional experiments, sequencing, fine mapping, sometimes in silico annotation, especially now with extensive data from ENCODE, etc., can be helpful, but often functional studies are also needed. And in fact, if you look a bit farther in this region, there are also two other interesting candidates. So the two genes I've highlighted here have been involved in congenital and hereditary forms of, heritable diseases, that involve some abnormalities of the white matter as well, so also potentially interesting genes. So what can be done to improve our understanding of the mechanisms underlying this association between this robustly replicated chromosome 17 Q25 locus and white matter hyperintensity volume? So on the genetic side, as I mentioned, you can do many things such as bioinformatics annotation, sequencing, expression studies, etc. But some help can also be given by refined analysis of the phenotype. And by examining the pleiotropy or shared genetic variation with other diseases, including other manifestations of small artery disease of the brain, and by examining gene-environment interactions. So how about the association of this locus with small artery disease characteristics? So is there an association with other features of small artery disease of the brain? So it is unclear to date whether this locus is associated with other MRI markers, especially the most prominent one, which are lacunar, I should say, small subcortical infarcts. So two studies have examined this association. Actually, the Japanese study that I referred to earlier and the Metastroke study, and they did not find any significant association. But the Japanese study did not indicate how they measured infarcts and what the prevalence of these infarcts was, so it's very difficult to conclude based on these findings. And the Metastroke project is a bit special because it was done, as I said, on ischemic stroke patients with all different subtypes of ischemic stroke. So people with MRI-defined small subcortical infarcts in this study probably had small subcortical infarcts of very heterogeneous mechanisms. So we have tried, and this is very preliminary data, but just a pilot study in the 3C Dijon MRI study, which is a large French population-based sample of non-institutionalized older people of 65 years or more. And we have found that in this study, so with about 1,600 individuals, and actually relatively small number of lacuna infarcts, about a small proportion. Actually, I think it's about 8 or 9%. But despite that, a small number, we found a significant association of the not the top SNP, but the second SNP, with an increased risk of lacuna infarcts, and also an increased risk of having extensive dilated perivascular spaces, which is another feature of small artery disease in the lenticular region. So that suggests that there may be, at least to some extent, some shared genetic variation between these two expressions of small artery disease, even though there may be some allelic heterogeneity. So this needs to be confirmed, of course, in other independent population-based studies. It is also unclear, whether this locus is associated with different white matter hyperintensity characteristics. Indeed, several studies have suggested that the risk factors and mechanisms of white matter hyperintensities could differ according to their location. So, for instance, whether they are periventricular or whether they predominantly subcortical can make a difference, and also whether they predominate in the occipital versus the frontal region. And some work from Yicheng Zhu and Anand Viswanathan, who's the assistants, has shown that in white matter hyperintensities related to cerebral amyloid angiopathy, white matter hyperintensities tend to predominate in the occipital region. And this was actually corroborated by the data shown by Dr. Kalimo this morning on the capillary changes that predominated in the occipital region. So we went on to look at this a little bit in the 3C Dijon study, and we found that both variants were associated very significantly with periventricular white matter hyperintensities and not that much with the deep white matter hyperintensities, and they did not seem to be associated with the occipital predominance, but rather with the frontal predominance, which could, again, need to be cautious in interpretation without replication, but it could suggest that the mechanism linking this locus to white matter hyperintensities may not be, or at least not largely mediated by cerebral amyloid angiopathy. And finally, the association of these chromosome 17q25 variants with white matter hyperintensity progression over time has not been examined yet either. And we did not find actually any association of the SNPs, and I'm just showing here because it was too busy otherwise, the association with the top SNP, but there was no significant association of the top SNP with white matter hyperintensity progression overall. But there was a surprisingly highly significant interaction with blood pressure levels, both with systolic blood pressure and diastolic blood pressure, and these were entered in the regression as continuous variables. And so then we stratified the analyses on the median value of systolic and diastolic blood pressure at the baseline. And interestingly, we found that the locus, the top SNP, actually it was mostly the top SNP, to a lesser extent the second SNP, was significantly associated with more a rapid extension of white matter hyperintensity volume only in individuals with lower values of systolic blood pressure and or diastolic blood pressure, while there was no significant association and even a trend in the opposite direction in individuals with high levels of systolic or diastolic blood pressure. So intriguing findings suggesting that there may be some modulating effect of blood pressure on the effect of these genetic variants or vice versa. So I've talked mostly now about this GWAS, but as you can see, these are very preliminary data. That's really the first step, and there still remain many unsolved mysteries on the small artery disease genetics, and Dr. Norving pointed out that actually results have been quite disappointing in other fields of small artery disease genetics as well. So where's the missing heritability, and what can we do to try to solve these mysteries? We had seen that the heritability is very high. We are explaining probably less than 2% of that heritability by the chromosome 17 locus. Probably the first step will be to do even more GWAS, because 10,000 is a large sample, but the latest GWAS on other continuous traits that are more easy to measure, of course, but such as height or weight, BMI or lipid levels were done on more than 100,000 individuals, so we're still far off from that. So a first step will be to, as much as possible, increase the sample size to increase power, and there's currently a second wave GWAS ongoing within the CHARGE consortium, which has doubled the sample size. It would be interesting to include non-European ethnicities because it can help approach the causal variant due to these differences in genetic architecture between ethnic groups, and to account for interactions with this very strong risk factor that is hypertension. So GWAS, actually I haven't said that before, but GWAS study mostly common variants with a minor allele frequency above 5%. So it is now recent studies have shown that rare variants also contribute to complex diseases, and it will be very important now to also explore the effect of rare variants on the small artery disease phenotypes. This can be done using sequencing, also some special genotyping of rare variants, and I will expand on those technical aspects a little more tomorrow. But just to let you know that there are such efforts already ongoing for small artery disease of the brain. And I would like to come back to my initial statement that candida gene studies had been quite disappointing, and I don't think this should be taken as a conclusion that they should never be done anymore. In fact, I think now we have very interesting tools with high throughput genotyping and to actually perform better and more comprehensive candida gene or pathway association studies that can be also very helpful in dissecting the genetic underpinnings of small artery disease. So for instance, a PhD student in the lab, Sabrina Schilling, has recently performed a meta-analysis of studies examining the association of ApoE with MRI markers of small cerebrovascular disease, and she found that overall there was a significant association of both the ApoE epsilon 4 and the ApoE epsilon 2 allele with an increased burden of white matter hyperintensities quite significantly, which is going in the same directions as associations observed actually with intracerebral hemorrhage, and the underlying hypothesis is that this is mostly mediated by cerebral amyloid angiopathy. There was also recently a very interesting study by the Graz group, Helena Schmidt and collaborators, that has shown an association of common variants in the Notch gene with white matter hyperintensity volume, and this was replicated in the CHARGE consortium. And finally, I would like to end by mentioning an ongoing project that is being carried out thanks to the Leduc Foundation, which I would like to thank very much, where we are exploring the contribution of genes involved in the pathways of cadazole and carazole in association with common forms of small artery disease. And this is being done by Ganesh Chauhan, who is a postdoctoral fellow in the lab. So my last slide is just to show that I've been focusing now on white matter hyperintensities, but of course it will be very important to get a full picture of genetics of small artery disease to examine also other expressions of small artery disease. So for MRI markers, we are currently within the CHARGE consortium performing GWAS of small subcortical brain infarcts, and perhaps this will lead to additional findings. Dr. Norving alluded to the studies that have been performed on ischemic stroke, and especially the Metastroke study by Matthew Traylor and collaborators on 12,000 ischemic stroke patients, which indeed has identified new variants in association with the cardioembolic subtype and the large artery subtype of ischemic stroke, but not at all with the small vessel disease subtype. So it's probably partly a power issue, and the heterogeneity of the phenotype and imperfection of the TOS subtyping probably play a role. And finally, intracerebral hemorrhage, there's very strong findings for the association with ApoE, the ApoE 4 and 2, mainly with the increase in size of the hematoma, which comes from the Boston group, Jonathan Rosen's lab, and Jonathan Rosen is also currently spearheading a large GWAS of intracerebral hemorrhage, which hopefully in the coming months will also lead to some very interesting findings. So I would like to end by acknowledging the people I've been working with on this topic, so the NeuroCharge Working Group, especially Dr. Seshadri and Dr. Fornage, the Department of Neurology of Lariboisière Hospital, Professor Chabria, Professor Bousser, the INSOM Unit 740, Professor Tony Lasserve and Professor Anjoutel, and the young investigators that talented young investigators that I'm very lucky to work with, and last but not least, Christoph Zorio. So thank you very much for your attention.